Great. So hi, everyone. My name is Shani, and I'm a PhD student at the Science Group. And today I will present one of my works in which I use machine learning for predicting bacterial sRNA-target interactions. So bacterial small RNAs interact with mRNA targets in different ways. Mostly regulation is exerted as an act of repression, as the sRNA either blocks the mRNA translation or leads to its degradation. In some cases, unlike microRNAs, sRNAs base pair with the mRNA targets to activate their translation. In this specific work, we predict whether an interaction will happen or not. So given a pair of sRNA and mRNA, our models predict and returns the probability that the two molecules will interact. And why should we predict mRNA-sRNA interactions? So today there are several experimental methods for identifying sRNA-mRNA interactions, but their application is technically challenging in practice. In addition, these methods are restricted to specific growth conditions. The current computational methods have a relatively high false positive rate and a low recall. Some of these methods are restricted to conserved sRNAs, and most of the machine learning-based methods were evaluated on a very small data sets or used a very limited set of features. With the growing amount of data collected in recent high-throughput experiments, machine learning methods gain more power and have the potential to outperform other prediction methods. And that is the focus of our work. So we use data of sRNA-mRNA interactions in E. coli that was gathered from various sources. The first data source, named SR-based, contains interactions that were manually collected from the literature. It includes both positive and negative interactions, as the negatives are sRNA-mRNA pairs with experimental evidence for having no interaction. The three other sources are high-throughput experiments that provided only positive interactions in the format of sRNA-mRNA chimeric fragments. To ensure the high quality of the data, we ran a pre-processing step that included filtering and alignment of the different data formats. Then we ended up with a unified dataset that's suitable for evaluating machine learning models. So the positive interactions for from the three high-throughput experiments were used as our train set. And to train a classification model, we needed to have some negative data. So we synthesized an equal number of negative samples by shuffling the sequences of both the sRNA and the mRNA fragments of each positive sample. The positive and the negative samples from the SR-based database were used as our independent test set. As part of our feature extraction process, we ran the RNA-up software to calculate a local interaction site for each sample in our dataset. We generate an interaction duplex and then extracted various types of features, including energy measurements, structure-based, and context-based features. We trained two machine learning models, XGBoost and Random Forest, and compared them to the state-of-the-art prediction tool named CopRNA. CopRNA predicts sRNA targets based on thermodynamics calculations and conservation across species, and it requires at least 3 sRNA homologs to provide a prediction. We evaluate our models in two ways. First, we ran a 10-fold cross-validation over the train set and measured 3 matrices to evaluate the performance of our models. Next, we compared our model's performance to CopRNA over an independent test set. Here, we used the pre-computed CopRNA predictions for 12 sRNAs that were available on Copra's website. In order to compare our scores to the pre-computed CopRNA scores, we had to set up threshold, define binary predictions, and calculate suitable evaluation matrices, which are different from the matrices used for the train set. The preliminary results of our work show that the Random Forest model succeeded to generalize better than the XGBoost model, and it achieves fine results in the 10-fold cross-validation over the train set. The results over the partial test set show that CopRNA is a bit more accurate than our XGBoost model, Looking at the precision recall trade-off, you can see that copper RNA is more precise, meaning it has a lower amount of false positives, while our XGBoost model has a higher recall, means it has a lower amount of false negatives. And both models achieve the same F1 score, which is close to 0.6. So these preliminary results indicate that the task of predicting siRNA-mRNA interactions is quite challenging, even when using state-of-the-art tools. However, there's still room for additional improvements to achieve better results. And this is what we aim to do in our next steps. So first, we plan to improve our models by using AutoML tools for hyperparameter optimization. We also plan to extract more features and examine more tools for duplex generation in addition to RNA app software. As mentioned before, the preliminary results were calculated over a partial test set, since we used pre-computed predictions of copper RNA. We have already started to run copper RNA to get its predictions for all the siRNAs and mRNAs in our test set. So when this is done, we will reevaluate our models against copper RNA using matrices that are suitable for comparing probabilities, such as ROC-AUC. At last, we plan to examine alternative methods for generating negative samples that may improve the training of our models. That's it, and thank you for listening.